going to do something that we're going to do in lecture again probably as well. I just wanted to talk about the anatomy of these nephrons and sort of their function in the creation of urine and the excretion of some of the more toxic chemicals that your body makes. So here's this sort of schematic picture of what the nephron looks like, and understanding how all these parts work is necessary. It's fundamental for understanding how the kidneys work. So how does the nephron form urine? And here's a very nice picture of it. It has to do with a few of these processes like glomerular filtration, tubular reabsorption, and tubular secretion. And I'm going to, instead of using this nice diagram, talk about them in cheesy, sort of hand-drawn, cartoony form. So here you have a very generic renal artery. It just looks like a red line, but this is artery, so again, it's taking blood away from the heart. And it's going to branch off into an arteriole, because remember, arteries branch into arterioles, and you'll see the arteriole is slightly smaller than the artery. This is not even to scale necessarily. The arteriole would be even smaller, probably. Especially if this is the renal artery, then the arteriole would definitely be smaller. But the blood branches off into this afferent arteriole and goes to this thing called, it goes to the glomerular capsule here. And so this is you're going to see some stuff starts to travel this way in a moment. Only smaller molecules are able to continue on in this nephron. So then blood, remember, came in with the afferent arteriole. It's now exiting via the efferent arteriole. And I'm just showing it sort of end there, or sort of shoot out into space, but it's actually going to go somewhere in a minute. So, like I said a moment ago, the formed elements are retained in the glomerular capsule. They flow into that efferent arteriole. And other things are filtered out into the next region. So this next region is going to be over here. This is called the proximal convoluted tubule, or the PCT. And like I said, large things like formed elements get stopped here. They keep going this way. And smaller things are going to go this way, up the PCT. And you can probably see why it's called convoluted tubule, because it takes a very convoluted path. And again, this is not necessarily anatomically correct, but it is taking this very convoluted path with this, what we call filtrate. And what's in the filtrate that's going to continue on into the PCT? Well, water is in the filtrate, urea is in the filtrate, there's some glucose in the filtrate, there's some uric acid in there, and there are salts as well. So all of these things being filtered out, continuing along their merry way on the PCT. So what does the PCT actually look like? We're going to take a closer look at it to kind of see a bit about its function. So on the right side of the screen, I'm depicting this is inside the PCT. This is going to continue on in the nephron. And then this is outside of the PCT. The stuff that winds up outside the PCT is mostly going to be reabsorbed by the blood. So inside the PCT, remember, this is that filtrate. The formed elements kept going along that afferent arteriole. But inside the PCT, you have water, you have urea, you have glucose, you have uric acid, and you have salts. But we want to keep some of this stuff. If you were to pee out sort of every time you drank water, you'd be pretty bad. You would not be well off. If you peed out every time glucose reached this point in your body, it wouldn't be good. And actually, you can do some urinalysis tests and look for high quantities of glucose to see if maybe there is a problem in maybe this region of your kidneys. So you have these transmembrane proteins, and that's why I kind of put them here in the middle between the two membranes, separating the inside and the outside. And some of them are pumps, so that's why this guy is a pump. And some of them are proteins for facilitating active transport. So really what these things are going to do is take stuff inside the PCT and push it across or against its concentration gradient to the outside of the PCT. So you'll remember that things typically only diffuse from areas of high concentrations to low. But because you have these pumps and you have these proteins here, you're going to be able to actually push things from a low concentration to a high concentration. But to do that, you need ATP. And so I put this little generator here. This represents the mitochondria. I'm only putting one because I put a bunch before it made it really messy, but you should know that these cells in the PCT have lots and lots of mitochondria. They also have lots and lots of what are called microvilli, so that they can expand their surface area. So cells in the PCT have many, many microvilli to expand their surface area, and they also have lots of mitochondria because they need energy to push all these cells against the concentration gradient. So remember what's in here. We've got some salt. We have some salt and we have some urea. We also have some glucose, which we'll deal with later. But you see the pumps are moving salt from this region of low concentration here to this region of high concentration out here against their concentration gradient. And that's all fine and good because, again, we have pumps, we have lots of mitochondria. But we also have lots of glucose in here. And that was not meant to suggest that the salt concentration outside the PCT just dropped. It was just to keep things a little bit cleaner. But with active transport, these glucose molecules are going to go to this transmembrane protein here. It's going to change conformation and push the glucose molecule out. All right. I don't necessarily need you to know the details of active transport. I just want you to know that the glucose molecule is going to go to this protein. This protein is going to change conformation and push the glucose out. It's going to use some energy to do this, which again, is why we have our ATP. So let's get rid of our last glucose molecule. It's going to change conformation so it can pick up this glucose molecule. It'll change conformation again so that it can push it out. All right, so we've gotten a vast majority of the glucose out. We've gotten a bunch of the salt out of the PCT. And so now, what's left in the filtrate? What's going to continue on in this loop? Well, water is still in there. There's still some water. There's still that urea from before. It's hiding up here in the corner, but there's plenty of urea in this side, the PCT. There's plenty of that urea. And there are also some salts left over, some residual salts that didn't get pumped out. I shouldn't even say residual. There's a fairly high concentration of salts that didn't get pumped out before. So now we've gone from this renal artery to probably a smaller branch, and then to this afferent arteriole through the glomerulus. Okay, blood continued on with all its formed elements out the efferent artery. the efferent arteriole. Then that filtrate continued on through the PCT and had a bunch of glucose removed from it and had a lot of salt removed from it. All right, so after it's done in the PCT, it's going to enter the loop of the nephron, which is also sometimes called the loop of Henle. And salt is going to come out. A lot more salt is going to come out. And you might be asking yourself, well, why on earth? Why would we want salt inside this extracellular space? Because that's what this is out here. This is this extracellular space. Why would we want salt there? Well, we want to increase the concentration of salt in this region because we want water to come out as well. We want to get rid of the least amount of water possible. And so this loop of the nephron, salt comes out. Salt is able to come out here because there are sodium pumps here. And because the salt comes out, there are what are called aquaporins. I don't need you to know aquaporin necessarily for this section, but there are water can easily come out of this side, the descending loop of the nephron. And you'll remember wherever there is solute, water is sure to follow. Okay, so the next thing that it does is it continues up this loop of the nephron, and what's left in here is there's some water. There's probably a little bit of salt in there, maybe a tiny little bit of glucose, but not very much, definitely trace amounts of glucose, and concentrated urea. So there's just a little bit of water left, enough to have that urea be dissolved in it, and and that's pretty much it. So then it's going to, this urine filtrate's going to filter into what's called the renal papilla, and then ultimately it's going to get squeezed into the urinary bladder and you're going to pee it out. So you might be asking, like, we had all this material sort of exiting the PCT into this extracellular space. You had water coming out of this ascending nephron. How does that actually go back to the blood? Well, I hadn't drawn it here, but this efferent arteriole continues out and spreads around into a ton of capillaries that are all around these different regions of the nephron. And so these capillaries are actually ultimately going to suck up the stuff that was pumped out or removed from the nephron. And again, this is definitely not anatomically 100% correct. It is not to scale. And then ultimately, this is going to go back into a venule, back into a vein, and then return to the heart to be, reoxygenated, do the rest of its stuff.